It is my pleasure to introduce Dr. Alexander Khoruts, who is the director of the University of Minnesota Microbiota Therapeutics Program, which is a multidisciplinary program that brings together clinical and basic science expertise and has been a major leader in the field of intestinal microbiota transplant. His group was the first to demonstrate engraftment of donor distal gut microbial communities into recipients suffering from multiple recurrent C. diff infections, and to develop the standardized protocols for donor selection, as well as to prepare cryo and lyopreservation microbiota. The program continues to focus its effort on the pharmacology and mechanistic aspects of this new frontier of therapeutics development. So without further ado, Dr. Khoruts, thank you so much for being with us today. We're very excited to hear your talk.
Thank you so much for the invitation. Well, I wish I was there in person, but certainly it's good to see a couple of faces that have their cameras on. Otherwise, I'll be talking to black boxes. So I'm going to challenge a number of paradigms. Let's walk through this. I have to have a disclosure slide.
I love that.
So these are the topics I'm going to try to cover in the short time. I'm going to talk a little bit about the science, even less about language, some about regulation, and maybe say a few provocative things about clinical trials. This is my index case. This is how I started in this field. Being at the mid-stage in my career at that time, being an immunologist at the bench, going to clinic once in a while, and seeing the emergence of microbiome science at the time, probably led by Jeff Gordon, with very interesting studies showing like transfer of lean microbiota into mice, or obese microbiota into germ-free mice would make them obese. And I thought, this is going to be the next big thing, but I wish I was 10 years younger or so. And then this patient came in, a 61-year-old woman who had chronic diarrhea for about 8 months to that point. It started with antibiotic treatment. She had a number of hospitalizations where she got hydrated and more antibiotics. She was quite miserable with bowel movements essentially every 15 minutes around the clock. Lived in a wheelchair, lost about 60 pounds of weight at that point. And of course, I did a flexible sigmoidoscopy right in the office, and she had pseudomembranous colitis. It was a unusual case. In fact, she was refractory to vancomycin and had to play with antibiotics at that time just to get it under control. And of course, everybody understands the basic bird's eye view pathophysiology of this condition. So as you all know, the colon is densely populated with microbial communities called microbiota. And anytime you use antibiotics, you're throwing a grenade in that and disrupt the local ecology. And that makes the patient vulnerable to invasion by C. difficile, which actually considers the colon its home, but it's gone renegade and it doesn't trust the host anymore. And so we ingest it generally in the form of spores. Once they find their way to the colon, the spores germinate and vegetative cells will produce toxins. Those will damage the lining of the colon, and that's the pseudomembranous colitis. And then we treat that with more antibiotics. And then if it's the first time, there's a 20 to 30% chance this is going to come back. And with every round of treatment, the chance goes up by another 20%. And after 3 relapses or so, you have an indefinite recurrent C. difficile infection condition. It's been known for a long time that there is a cure, and you could just simply put the microbes back, fecal slurry, and that recovers the ecology. In Western medicine, that's generally attributed to Dr. Ben Eisman's team in Colorado, published in late 1950s. In around that time, there was a flurry of these kinds of procedures done, including here at the University of Minnesota. Dr. Wangenstein did it. And then after a couple of years, it kind of died out because vancomycin was introduced. This was way before C. difficile was identified as the cause of this problem. But vancomycin, it actually was thought to be caused by Staphylococcus aureus. As you know, vancomycin covers that. So the problem kind of went away, not completely because there was a trickle of these case reports of people still doing it. But essentially, it was way on the periphery of medicine until C. difficile changed and everybody was facing this problem in their clinics, including a part-time clinician at the time, like myself, who ran into this patient. And so we did this. Being a scientist, I thought, oh, if we're going to do this, we have to do some science on this and actually demonstrate that the hypothesis that what's being done is recovery of the microbial community structure is what actually happens. And so we did that, and it was a case report published in Journal of Clinical Gastroenterology, picked up by the New York Times, and we really made a big splash with the New York Times part, not the JCG. And for about a year, we were heroes in the scientific community because we did shake a paradigm. It was thought in the science world that once your microbiome is established, it's very difficult to shift that to anything else. This idea is no longer quite there, although there is some validity to it, as I'll return to later. But then that was a brief celebrity because this was really picked up way too far, and people started thinking this is a cure for everything, and that generated quite a bit of legitimate resentment in the scientific community. Anyway, this practice picked up fairly quickly, especially after we published a method to standardize donor selection and cryopreserve the microbiota. So that was published in 2012. Within a month of that publication, OpenBiome decided to have a business, and that really accelerated uptake of this procedure, especially because this was a big clinical problem, with about 500,000 people in the US getting these infections and about 100,000 getting into this multiply recurrent C. difficile infection. So the FDA took notice of this and had a workshop back in 2013, politely listened to the scientists who expressed on day one, who expressed a lot of skepticism, and then to clinicians on day two who expressed a lot of optimism, and then came out with this prepared talk where they said, well, okay, what you're doing is illegal, because when something is used to cure, treat, mitigate, or prevent a disease, then it's called a drug. And this stuff is a drug and a biological product. Now, there was debate about this. The key word, because of course we know that liver transplant is not a drug, for example. So what is the distinction? And they pointed out to the language as written by Congress that transplants refer to human cell or tissue or organs. And this material is not human. Therefore, the only other category for this is a drug. And that's essentially how it is in the United States. This is, has been a matter of some debate around the world. It found an intermediate regulatory category in Australia in its own slot. And I understand the Europeans are actually leaning toward being convinced by the arguments that the human microbiota actually is human, a part of the human body. Anyway, what that meant is if you're going to use a drug that's not approved, you have to do it under an IND, investigational new drug application, which is a pretty laborious regulatory barrier. And there was an uproar of in the community from patients and physicians that this was governmental overreach. And so within 2 months of that workshop, the FDA came out with an enforcement discretion policy. Essentially, it is a don't ask, don't tell. As long as you're treating C. difficile infection that's not responding to standard therapies. Of course, you have to do informed consent about it. And that's been the rule in the United States ever since. Probably to be changed as soon as we have approved products. So I want to show you a couple of these kind of microbiome overviews. This is what a microbial community looks like in a very oversimplified way. As you can see, as you all know, there are hundreds of bacterial species. It's a quite organized community. These species are adapted to the human host. And of course, they interact with each other. Some of these interactions are mutually beneficial. Some are competitive. So this is a network analysis. The green lines show cooperation between different microbes. The red lines show competition. And of course, this is woefully oversimplified, not just because it shows too few bacterial species, but also because it's missing fungi, it's missing archaea, it's missing viruses. For every bacterial cell, there are 10 bacteriophages. And all of that makes for the microbial community that we cannot claim to fully understand by any means. However, this is what a microbial community looks in a patient with recurrent C. difficile infection. Essentially, it's been decimated. If you think of a community of any kind, let's say human community, then there is all kinds of people that are needed to make for a functional society. You need construction workers, you need hairdressers, you need a couple doctors, you need some lawyers, etc. This is what it looks like if all you've got is lawyers, for example. Not a very functional microbial community. Within a week of transferring an inoculum of fecal material into a patient like this, you see a essential recovery of the entire community. So you have to really register this picture in your mind whenever you think about using probiotics, for example, as a solution. You simply cannot restore a complex microbial community with a couple microbes, let alone that those products don't really belong in your GI tract because they've been in the lab for 100 years. But I've been fighting to some extent this idea that it's not human transplant. It had something to do with the name of this. It was Larry Brandt who came up with a question of how should we call this, because this was called fecal bacteria therapy. And he asked the question, it's more than bacteria, right? So we really struggled with the name. And so, well, you know, microbial community is called a microbiota. And what we're doing is a kind of a transplant. And I really wanted the transplant paradigm there so we wouldn't go down the drug pathway. And so the microbiota transplant stuck. And then people added the fecal work to it, and I'll return to that. Well, anyway, if it is a drug, and since this debate is essentially over, I've accepted it. And then, but the next question is, well, if it's a drug, what is its pharmacology? And pharmacology has a number of branches. We mostly talk about the pharmacy, which is the formulation, what goes into it. If you're going to do cryopreservation, you have to think about specific cryopreservatives. If you're talking about lyopreservation, which is freeze drying, then there's lyoprotectants, there's encapsulation, there's a lot, and there's donor selection for the active ingredient. There's a lot that goes into pharmacy, but once you're done with that, you want to ask something about pharmacokinetics and pharmacodynamics. Pharmacokinetics is essentially a study, in this case, of engraftment, and unlike other drugs, which you need to continuously give, this may be the case here too, but what's different about live microbial therapeutics is that they expand. And the typical measurements that we do, of course, don't apply here. And you have to incorporate concepts and technologies from microbial ecology of how we study this. And if you want to understand how this works, then you have to think about pharmacodynamics. What is the change in metabolites or whatever else that is mediating the curative function and perhaps toxicity? And if you understand those two, then you come up with pharmacotherapeutics, where you know what your regimen is. And of course, you have to incorporate understanding of toxicity, which in this case, it could be, for example, some pathogens sneaking in, and drug-drug interactions. It amazes me how commonly referring physicians don't really think through that combining microbiota transplant with antibiotics makes no sense whatsoever. So for that patient, this was the technology. Pretty self-explanatory. I discovered at the time that the O-ring here is an essential component of methodology. You also need a couple additional ingredients with that old way: determination and courage. And it worked wonderfully, except it's not really mainstream medicine. What I was fortunate at the time is when we opened up an endoscopy unit in the suburbs, and this is where I volunteered to go, and no patients were coming. And so I simply boarded off half the endoscopy unit, or actually the entire endoscopy unit, just for this. And so when the blender blew up in my face and the nurses were running around with air fresheners, this didn't terminate my privileges. But it was really not mainstream science. We took it in the lab after 10 cases or so. And this was the original preparation, as you see in 2010, fresh slurry that's been filtered. That still was quite impractical. And we started playing around with how to cryopreserve this material. And so in 2012, you see the middle slide or middle image of what it looked like in 2012 and still does. This is frozen with glycerol. It's filtered fecal material. Each dose is quantified in terms of numbers of bacteria. While the field is still talking about grams of stool, that's really not the way to do it, because the the burden of bacteria or presence of bacteria varies per gram of stool between individuals by about 10-fold. So we count them. The unit contains generally 5 times 10 to the 11th bacteria. And then we started working on encapsulated preparation. And by 2015, we had something that seemed to work. And by 2018, we came up with a serious innovation, which is we made our capsule Swedish orange color, which really does enable placebo-controlled trials. All of this done in this unit here, all but the donor program, where we manufacture these products using good manufacturing practices protocols, which is the pharmaceutical standard if you're going to do this as a drug, or even if it's a blood transfusion. So the elements of this program is that, of course, there's a donor program. And I'm happy to talk about the details of that. But essentially, these are absolutely healthy individuals who are rigorously tested in all aspects of health. They're all lean, they look like Greek gods, and they have no diseases, no prescription medications. And these days, we have to monitor them for COVID, so there is a daily diary and a frequent testing according to a schedule for COVID. The material is delivered to the production suite in this facility and processed within 2 hours. And there is a very standardized way of doing it. There are some key steps where several people have to sign off. And in the end, each lot of material has a batch record, and that goes through an independent quality assurance and material release process. That's basically how any pharmaceutical operation works. Again, with the donor program, there is, of course, initial screening. It took me a couple of years to build the donor program so we could recruit people and fight some, I guess, prejudices at the time. But these are committed individuals who understand that they are really partners in this. There are some inclusion-exclusion criteria, which are quite rigorous, and 90+% of people are not qualified. And that's, of course, depends on the pool. You don't go around hanging posters that you need some stool donors, because you're going to get some really weird people coming for that. After that, as it goes through the production process, there's also release criteria. So it's still possible to fail, and then the material is destroyed. We have retained samples, of course, as part of the process in case we ever have to go back and recheck for something. And this gives you an idea why there was such pushback from the physicians of operating under an IND. This is just an amendment to a protocol that we put together back in 2012, describing the capsule modification. The original one weighed 22 pounds and had to be mailed in 2 boxes by FedEx. Now, let's think a little bit about the pharmacology of what we're trying to do. So on the left-hand side is the typical of what we do. A drug gets absorbed, it goes through the liver, there's some intrahepatic circulation, some of it may be pumped back out, for example, some of it is modified, gets into circulation and goes to the target organs. We don't think that much about what's happening in the intestinal compartment, but it is its own compartment, of course, and it is intimately open to the environment. Again, there is a very dense microbial community there, highly adapted to its host, quite integral to the host physiology. Louis Pasteur, for example, did not think that germ-free animals could be viable. He was proven wrong, but germ-free animals are not normal. They're about 40% lighter in weight. If you're talking about mice, they don't have a mature gut. They're extremely susceptible to infection. Doesn't take much to kill them over. And you all know this because there's multiple functions that these microbes do, not least of which, of course, is protection against invasion like C. difficile. This microbial community is not static, it's dynamic. It's modified in real time by the nutrient flow that comes in. There could be pathogens around, and in some way, we all share this community also among each other. Recently, it's been shown, for example, that the horizontal gene transfer in industrialized nations is much greater than that in developing countries for probably multiple reasons, but suggesting that we all kind of share this a little bit. And if you think about pharmacokinetics, then you might recall there's ADME parameters: absorption, distribution, metabolism, excretion. Distribution, for example, would suggest that we're particularly interested how these microbes distribute along the GI tract. We know the colon is where most of them are, but even within the colon, some are in the lumen, grazing on the nutrients. Some are adherent to the mucosa, and so mucosa adherent microbiota are somewhat different from what you get in the lumen or in the fecal material. And there are subcompartments, for example, the microbes that may interact with the crypt stem cells that could be really important for colon cancer are different than the ones that tickle the tip of the villi, etc. Generally, what we study is fecal material because it is what's most available, but that's used as a surrogate for everything else, and those studies are sorely lacking. So this is a kind of a pharmacokinetic curve of what happens with our capsule preparation. So before the transplant, this is kind of what it looks like. So I'll just walk through this a little bit. This shows the phyla of bacteria. The green band is Bacteroidetes, and the blue band is Firmicutes. Those are the dominant obligate anaerobes, dominant phyla in the colon. Proteobacteria, which we're most familiar with, that contain microbes like E. coli, Klebsiella, Proteus, and others, are a small minority normally, but they are massively expanded in relative abundance in these patients, mainly because they are treated with vancomycin typically, which hits both of the dominant phyla. Within days of this capsule administration, you see shrinkage of proteobacteria, expansion of Firmicutes. There is some delay in restoration of Bacteroidetes. This black dot with little error bars shows the diversity index. The Shannon diversity index is made up of richness, which is the number of species being detected, as well as the distribution. So, or evenness. So if you have, for example, one or two species that dominate, even though you can detect hundreds of others, that's not very even. All right, so I guess the point of this slide is it takes like 3 weeks before you stabilize in the microbial community after a single administration of this capsule preparation. And then we're trying to understand, well, which bacterial taxa may be more important, and can we identify a pattern that would be predictive of response or failure. So typically, if it's going to have a failure, it's going to happen somewhere in the second week after you stop the vancomycin. But we can predict who is going to fail at about a week. So before the symptoms happen. This is a complex slide. So the crosses, the red crosses here, Oh, first of all, we're looking at a PCOA plot. So each dot is a microbial community. The distance between each of these symbols is how different they are from each other. It's a multi-dimensional space. We're only showing two axes which show the most difference. The red crosses is the microbial communities of the pre-FMT patients. Within days, and the donors here are the black dots here. Within days, you see some movement toward the donor, which are these green circles. Within weeks, more movement toward the donor. And then within months, you see essentially stabilization there. If you look at specific taxa that are predictive of success or failure, it's Lactosporaceae, Ruminococcaceae, and some members of the Bacteroidetes of phylum. Not surprisingly, you see a shrinkage of Enterobacteriaceae, which is the proteobacteria. Now, to really understand a little bit more about what should we be looking at in pharmacodynamics, you have to know a little bit more about C. difficile pathogenesis. And there is a lot going on. It's a very smart pathogen. And one of the, so this is a schematic of what is normal homeostasis. So what you see normally, of course, is a healthy mucus layer. You see nice tight junctions between the epithelial cells. Those work to maintain the gut barrier. The microbiota is generally compartmentalized away from the host, so there's not too much interaction. And the immune system is right behind, which is integral to the mucosal barrier, with IgA being pumped in and saturating the mucus layer, and all the different cytokines maintaining the integrity. Over on the microbiota side, in the colon, one major part in C. difficile life cycle is that its C. difficile senses the composition of bile acids. With secondary bile acid metabolism, you have moieties like glycocholic acid and deoxycholic acid dominating the colon milieu, which tend to be inhibitory to C. difficile spore germination and vegetative growth. In addition, the other microbiota can make other compounds like tryptophan and indole-derived compounds, which are inhibitory to the growth of C. difficile. When you take antibiotics, what happens is, of course, you have fewer microbiota, but that also you have a less of a mucus layer. The microbial signals actually provide tonic stimulation to the epithelial cells, which enhances mucus production as well as tight junctions, etc. As was mentioned in just before this talk with the discussion on enteral nutrition, this could very well be in play, that when you starve the patient by giving TPN, and maybe you throw some antibiotics on top, you really are messing with the gut barrier and making the patient much more vulnerable to translocation by microbes that don't really care that much about the host. Also, what happens is the secondary bile acids in the colon are no longer present. The secondary bile acid metabolism in these patients is essentially knocked out. And you have primary bile acids like cholic acid, which is actually used in the laboratory to germinate C. difficile spores and grow C. difficile. All right. And then C. difficile, as I said, is a smart pathogen. Once it senses that it has an opportunity, it's going to make quorum signals of its own. It's going to secrete, for example, proline-based cyclic dipeptides, which are going to further inhibit the microbes that mediate secondary bile acid metabolism. And it's actually going to collaborate with other microbes like proteobacteria, which will further augment this inhibition through production of certain indoles. And as these quorum signals will also transition the state of C. difficile to a stationary phase and get it ready for invasion. So C. difficile can go from flagellated and motile to stationary, which tends to find to translocate better. And you'll see some sporulation. It's getting ready for potential hibernation. There is an idea that C. difficile can also hide in biofilms in the mucosal tract. It's another way of maintaining stealth existence. Once the C. difficile translocates, the toxins actually, the enterotoxins that it makes have receptors on the basolateral side. It causes necrosis of epithelial cells, and you have translocation of microbes, which is met by neutrophils and other immune cells. And when you see pseudomembranous colitis, you're essentially seeing the immune defense. What happens is neutrophils literally explode, spill their guts, throw their DNA as kinds of nets. In fact, this is called neutrophil extracellular traps. These pseudomembranes provide temporary patches in the break in the mucosa. Early on, we looked at metabolomics of what happens in the course of the microbiota transplant in these patients, and basically stumbled in the bile acids as the major differences. So patients with multiply recurrent C. difficile infection, as I mentioned, have very little in terms of secondary bile acids like lithocolic and deoxycholic acid, but they have abundance of taurocolic acid, for example, which is a progerminator. That is rapidly reversed after the FMT. That may be one mechanism of how it works, but I am certain that it's not the only one. The second topic I'd like to mention briefly is the language we use. This is an area of cognitive linguistics. Language shapes how we view the world. As I mentioned, the term fecal microbiota transplant came up in 2011 when Dr. Brandt raised the question how we should name it. This group had a discussion. I came up with microbiota transplant. We had a brief debate about the word fecal. Everybody felt, other than me, that was the right way to describe it because that's where the microbiota comes from, and this is how we should communicate with patients. What happened, of course, as you all know, is that nobody uses the word microbiota and became fecal transplant. I'm here to say that there is no such thing as a fecal transplant because feces is a very complex substance and you simply cannot transplant it. What you're transplanting is microbiota. And I got sensitized to that when I heard an industry talk showing disgusting feces and then showing their aesthetic product in a capsule or in a liquid formulation and showing this is the future, which of course is not entirely fair. So after brainstorming with Dr. Brandt, we came up with an alternative, which is intestinal microbiota transplant. We really went through different words. I still thought the transplant was a good name because it highlights, in this case, that there is a donor involved. There is an actual human donor, and there is a donor program, and this material is live, so that's distinct from implant. This really is a transplant. We're transplanting microbiota, but by simply switching from fecal to intestinal, we focus on the recipient rather than the donor. That was published even made it in the Schlesinger textbook, but I don't hear it picking up. And of course, it may be too late, but another weighty argument is that IMT doesn't quite have the same zing as FMT. So you can't really legislate language. My preferred term actually now is microbiota transplant therapy, because things can be quite more complex than simple administration of one time and you think you've really changed the microbiome to that of the donor. That really is disease by disease case. And so I've decided, okay, I'm going to give up on this battle. We're going to name our preparations like drugs, like the drug companies do. MTP stands for Microbiota Therapeutics Program. This would be a 101. This is a C for capsule. This is a liquid. R for a recurrent. We have different doses for recurrent and fulminant. And there could be different formulations. In fact, we already have 102 and possibly 103. Usually there is a question about probiotics. So I thought I would throw this in so people really understand the difference between drugs, dietary supplements, and foods. We talked about drugs. This is something that is used in diagnosis, treatment, mitigation, cure, or prevention of disease, and requires, of course, approval by the FDA. Then we have dietary supplements and foods, most of which have structure function claims. And you usually see a disclaimer: this product is not intended to diagnose, treat, cure, prevent disease, even though the physicians often recommend stupid things like take probiotics after you take antibiotics to prevent some disease. It has nebulous terms like promote digestion, which I'm not sure what that means or exactly what tests you need to do for that, but it doesn't require evaluation by any regulator when you're that nebulous. You might have some health claims by certain products, which is reduction of risk of disease in a healthy population, like use of calcium in the diet on a regular basis may help to reduce the risk of osteoporosis. This is somehow sneaked in. There is some science there, and that requires pre-approval by the FDA. So you really can't equate probiotics and these microbiota therapeutics in the same category. We're just completely different. And we need to think about the appropriate scientific framework if we're going to talk about appropriate regulation. And this is, of course, a paradigm that is perhaps the most foundational to medicine. It's the germ theory of disease. And here you have Koch's postulates where the focus is on a specific pathogen, for example, C. difficile, where you go through these requirements in order to say, yep, this is the cause of the disease. It must be observed in every case. You can isolate it in culture. You can inoculate animals. You can reproduce the disease, and you can recover disease from diseased animal. Okay, so the focus of infectious disease medicine has been mainly on the pathogen, with essential ignorance of what happens to all the other microbes when you're treating that. And all the other microbes that require thinking about microbial community. And you have to think in terms of ecologic principles. And in fact, it's the entire microbial community that now may be working in it by a different mechanism and driving a number of diseases, whether it's obesity, autism, or whatever else. 10% of metabolites circulating in your blood are either manufactured by microbes or are modified by microbes. The microbiota really is an organ in the human body, even though this organ is obviously distinct from others, being completely open to the outside world with which it has an intimate interaction. The, so to go back, sorry. So currently, the regulators are really essentially focused on what goes into the formulation. So, everybody may have heard about potential pathogens that could be transferred, and that's certainly a risk. But the proper way to think about it is not just what goes in, but what is the result? So the question is still open. When you're treating a patient something like C. difficile infection, where somebody has a decimated microbiome, and let's say you come up with a formulation that's made of a couple microbes that may act as a scaffold or may somehow inhibit C. difficile for just long enough, and then the patient builds their microbial community on their own, what is the ultimate outcome? Does the microbial community look like in a couple months or in a year? What is the burden of antibiotic resistance genes? What is the burden of different virulence factors? That is yet to be studied, but should be with every preparation. The final paradigm that I'd like to approach a little bit is how we think about how science actually works and how we fund it. This probably is the most provocative part, but it's certainly occupying a lot of my time. Normally, we think of it in this linear fashion. Somebody has an idea at the bench, goes through basic research, then it goes through phases of clinical trials, and then there's regulatory approval, and you got patient care, right? This is the way most people think about it. This is the way the regulators think about it. This is the way the NIH thinks about it. Of course, the reality is something like this. It's certainly well illustrated by what happened in this field, where the initial observations were made directly in patients, and then we went to the lab. And in fact, even though most of my grant funding comes from studying mice, and very little for grant funding goes to study patients, most of our impactful work, of course, and most of what we learn comes directly from patients. In this case, because human microbiota really is different from mouse microbiota, and mouse is a very different organism than human, for that I can go into many details on. But in general, this is a circular process. Even in an example of COVID vaccines, you could say, well, actually the first time people came up with vaccines could be, you could name Edward Jenner, for example, with a human experiment. And then there were decades and centuries of back and forth between. But this is the way the funding works. And in this case, with this product, we have a problem with this death valley of how do we proceed to transfer ideas that come from the academia or physicians or scientists to the companies that work toward commercialization. The paradigm says that the taxpayers and the society will fund the basic ideas, and then it's the job of industry to take them over and bring them to commercialization. Problem here, particularly, what is the intellectual property? This rich microbial community in a transplant may be actually the best possible option to reconstruct somebody's microbial community, but it has very weak intellectual property. And so you see investors really thinking, what is the product that will give us the most of that so we can charge the most money and reward the investors? It's very legitimate in the way it operates. And I think we do need to question how well this works. An example here that I work in cystic fibrosis, for example. In my research is on colon cancer and cystic fibrosis specifically, but I've been fortunate to work with this patient community. And you look at the trajectory of what happened in this field. 1950s, this was essentially a fatal disease in early childhood. And there were a number of important steps in diagnostics and therapeutics. And today, a patient with cystic fibrosis can expect to have a median life expectancy of 42 years. And if they make it to 42, they can expect to live another 20. And newborns today with cystic fibrosis are basically told so there's a good chance you're going to live a normal lifespan. There's only 30,000 cystic fibrosis patients in the United States. What makes this community kind of special is that they all banded together, created the Cystic Fibrosis Foundation, and funded a lot of the research that went into this. Drug companies were not really interested in this disease. The market is small. Once they did do some foundational research, they were able to cooperate with the drug companies, and cystic fibrosis has $1 billion in research funds and spends per patient on research certainly more than 100-fold relative to inflammatory bowel disease, which is 3 million patients in the United States. And with that, I'll say what the missions of our program are. So I believe we're the only program in the US that is manufacturing these products by GMP methodology. Our mission is to cure patients with C. difficile as the primary mission, or the original one, and we treat them free of charge. The other huge mission is to support academic clinical trials. So I talked a lot about C. difficile. However, you cannot translate easily the experience from C. difficile to other diseases without understanding the pharmacokinetics and pharmacodynamics implied. For example, in ulcerative colitis trials, we've seen a consistent theme that you need to do repeated administrations. That's because it's much harder to shift the microbiome in ulcerative colitis to that of a donor than in C. diff, whose microbiome has been decimated. A typical C. diff patient had 5 months of antibiotic conditioning, for example. There could be a very different choice or microbiome-informed choice in donor selection. So we actually do have an ongoing trial in ulcerative colitis that's placebo-controlled using daily capsules for 60 days. With, of course, the results are not in yet. My research program is mostly focused on trying to develop the pharmacology of this field, and that means engraftment studies. It means some metabolomics. It means really understanding pharmacodynamics. We continue to advocate for appropriate regulatory policies and promote education. And really, the main funding for this, as I mentioned, isn't coming from NIH, although we have had some, and we have some from Department of Defense. But mainly it is coming from trying to organize patients to be advocates for themselves. So we do have a nonprofit that is Achieving Cures Together that has been essential to this work, and I want to acknowledge them the most. I'll open up to any questions if you have any, and thank you again for the invitation.
Thank you very much. It's you've had an incredible journey following the clinical questions and the science. So where do you think the most innovation will come from? Will it be from consortia that are developed, microbial consortia developed by industry or academia? administered as pills? Do you think the innovation will be something along the line of metabolic products of the microbiota? Where do you think it will actually end up in the end, if not stool forever, which I think everyone agrees is not the thing forever?
I think the stool is already passé. While the FDA still allows blending raw poop and putting it in, it's never been mainstream, and it never will be, and for good reasons. So, our product, the lead product, the capsules that contains the full dose, is quite purified bacteria that do come from stool. And it's composed of 3 to 5 capsules, depending on the lot.
But myriad organisms, probably.
So the question really, yes, it's as complete a microbial community as we can get. And our goal is actually not to reduce it too much. Some of it happens unintentionally in processing. That is a debate. So you have on the one hand side, there's the reductionist scientific view that we can come up with a consortium that can bridge you to recovery in C. difficile. Or we can just give you the full microbial community in this disease. And I think the answer has to be in clinical trials that look at long-term outcomes, including looking at what actually happens to the microbial community after this treatment. And my bias is that if you give the whole community, you'll be better off because you've done the most at screening what you're putting in and testing what you're putting in terms of pathogens. If you just give a little scaffolding and let the patient who may be hospitalized a couple of times a year to build up the microbial community on their own, it very well could be that the end result is a much less healthy microbial community.
But that approach probably has greater regulatory challenges than a defined selection of bacteria that is reproducible time after time.
But it's reproducible on what you put in. It's totally not reproducible on what comes out. So in my opinion, what the regulators have to break out of the paradigm of just thinking what goes in, but also, that's important what goes in, but also think about what results, right? That's ultimately we care about the patients, not about the pill. And ultimately we care about the patients, not about the investor. The investor, of course, wants intellectual property. And the scientists, so I will admit that I'm in the minority view because most of the scientists do think like you, and they're really attracted by the reductionist approach that we are so smart that we can come up with a consortium of microbes that will be superior to what nature has created. That could be, it's yet to be demonstrated. You can say, well, what happened to breast milk? We had formulas for decades, and we're still not quite as good as that. So I'd like to see the actual demonstration that they're better. That doesn't mean that these products, the so-called full microbial consortia, cannot have intellectual property. I mean, we can look at microbiome diagnostics. We can select perhaps different enterotypes that may be, for example, more applicable for specific applications like ulcerative colitis or autism or something along those lines. And that's kind of where my research is trying to look at that. Or you can go full-blown investment into consortia, and that's absolutely happening. It's just the science will have to show that. We can't just box ourselves in with just our, preconceived notions. Just because we can see a small part, we don't see the whole universe. You need to think about it holistically. That's at least that's my view, and there's many more who will advocate the opposite.
I'll ask a quick question, thinking about it holistically in terms of, in addition to the microbes that we generally focus on and even read out in terms of changing in the microbial communities, do you think any of the other factors that are in these preparations, such as the fungi, the archaea, the bile acids, metabolites, are playing a role in any of these treatments?
I don't think, for example, in our preparation, the metabolites play any role because it's been so, the microbes have been so separated away from the fecal material. When you condense all of that to just a couple of capsules, and if you really think about what's in the capsules, there's more than half of it is the lyoprotectant that keeps the microbes alive. So I don't think it's the metabolites that are in the preparation, but it's certainly metabolites that microbes make. As far as other components, so yes, we do quantify the preparation in terms of the numbers of bacteria, but there's other domains of life there. We haven't been particularly good at looking at fungi. That's still an emerging field. They could be important. There is, of course, the idea that bacteriophage is a critical part of this and may have independent therapeutic function independent of bacteria. So there is a trial ongoing of using sterile filtrates that presumably contain just bacteriophage that may have therapeutic properties as well. And that's part of the microbial community is you have predators, you have prey. In this case, bacteriophage is predators, and they're probably integral to how the microbial community forms. I think it's extreme arrogance to think that we are so smart at this point to be able to construct microbial communities that are good for us from basic principles. We can't even predict what happens when you release wolves into the Yellowstone Park in macroecology. And what makes us think that we can do that in something much more complicated as intestinal microbiome?